This is the second part of the muscle lecture. This model is used to divide the regions of the muscle tissue up to see where the muscle fibers change shape and size when a muscle contracts. The Z-discs are a zigzag of dense material separating the sarcomeres. The A-band is the darker area between the Z-discs. The H-zone is in the center of the A-band and is the light-colored area. The I-bands are located to either side of the A-band and are also a light color. So here in the center you have, or on the ends you have the Z-disc, you have the A-band, you have the H-zone in the middle, and you have the I-bands here on the ends. These different zones and bands are created by the partial overlap of myosin and actin filaments within a sarcomere. So previously we talked about these fibers running lengthwise. You can see here that these run this way lengthwise, these run this way lengthwise, but they're not continuous. They're going to have breaks in between each section of the muscle. Myosin is a golf club-shaped protein and is the thick filament. This filament forms the cross bridge when a sarcomere contracts. Actin is one of the thin filaments and is helix-shaped. It is also anchored to the Z-discs at the end of the sarcomeres. It contains the myosin binding sites. Troponin and tropomyosin are both thin fibers that cover the myosin binding sites on relaxed muscle. When troponin and tropomyosin are covering the myosin binding sites, myosin cannot form the cross bridge and the sarcomere cannot contract. Sarcomeres shorten and muscle contracts using sliding filaments. The myosin head of the thick filament pulls on the thin filaments. The thin filament will then slide towards the center of the sarcomere. The thick fibers slide past the thin fibers. The filament length does not change even though the fibers slide past each other to shorten the sarcomere. While reading this and listening to it, it probably sounds pretty confusing. That's why we're going to watch a short animation here that is going to show this process and help clarify it.
In a relaxed muscle, actin and myosin myofilaments lie side by side, and the H zones and I-band are at maximum width. During contraction, the actin and myosin myofilaments interact. The actins slide toward the center of each myosin myofilament. As a result, the sarcomeres shorten. In the fully contracted muscle, the ends of the actin myofilaments overlap, the H zones disappear, and the I-band becomes very narrow.
So in this picture you can see where you've got your Z-discs, you've got your filaments partially overlapping, and as they contract your overlap is going to be much greater. This illustration shows a comparison of a sarcomere with different bands when it is relaxed and when it is contracted. Notice how the individual filaments do not change length, but just slide past each other to create more overlap and shorten the sarcomere. This animation shows the muscles contract. This animation shows the muscles contracting. It's going to show this muscle on an insect, but it is going to be the same thing in a human muscle fiber. So we can see the different muscle fibers or muscle cells within the skeletal muscle. And the different myofibrils within the muscle fiber. And then we have each of the sarcomeres next to each other. And then we have the different bands and zones. Sometimes the terms H-band and H-zone are used interchangeably. So we have the actin filaments here. And the myosin. And they're anchored to the Z discs or Z bands on the end. So this shows a sarcomere contracting, the filaments sliding past each other. And when we have multiple sarcomeres next to each other, each sarcomere shortens a little bit to shorten the entire muscle fiber. So when we enlarge it here, we can see that the actin is a little bit more helical shaped, the myosin a little bit more golf club shaped. And then again we can see the sarcomere shortening here. And in this scene it enlarges it a little bit more. And this is showing the ATP involved in the contraction. We will discuss this portion a little bit more later on in the lecture. So how do our muscles know when to contract? The information is transmitted at the neuromuscular junction where the nerve meets the muscle. So how do our muscles know when to contract? The information is transmitted at the neuromuscular junction where the nerve meets the muscle. The action potential is the electrical signal or stimulus that carries the message through the nervous system to the target. The motor neuron is the neuron which delivers the stimulus to the muscle. The motor unit refers to a single motor neuron and all of the muscle fibers it stimulates. The size of the motor unit varies with the precision of the movement. The motor endplate is the specialized region of the sarcolemma that connects to the nerve. There is a slight gap between the axon end and the motor neuron and the motor endplate called the synaptic cleft. The events that occur at the neuromuscular junction are referred to as the excitation of skeletal muscle. The electrical impulse is not able to cross the gap at the synaptic cleft, so it must convert it to a chemical signal and uses acetylcholine, which is abbreviated as ACH, to diffuse across the space of the cleft. When acetylcholine binds the motor end plate, we have activation. With activation, we have the sodium ion channels open. The flow of the sodium ions across the sarcolemma generates the action potential, which will spread along the sarcolemma. In order to be able to have the muscle relax, we must remove the acetylcholine from the cleft to close the sodium ion channels. Acetylcholine esterase will break down the acetylcholine remaining in the synaptic cleft. This is an animation showing the neuromuscular junction being stimulated. So here you can see the propagation along the nerve. And you saw it being released into the presynaptic area. So this is the presynaptic neuron enlarged, the synaptic cleft, and the postsynaptic area. This link is available to you in Angel, so you can see the effects of certain snake toxins, pufferfish, dart toxins, and spider toxins if you're interested. This animation here shows the synaptic cleft. When you start the action potential, you can see the electric impulse come down, releasing the acetylcholine into the cleft at the vesicles, binding the motor end plate, and then opening the sodium channels. That will depolarize the muscle cell and trigger the action potential. The events that occur at the neuromuscular junction are the excitation of skeletal muscle. So the electric impulse is not able to cross the gap, so it will use acetylcholine to cross that space of the cleft. The acetylcholine binds the motor end plate for activation. That will turn around and open the sodium ion channels, allowing the flow of sodium across the sarcolemma, generates the action potential. In order to be able to have the muscle relax, we must remove the acetylcholine from the cleft to close the sodium ion channels. Acetylcholine esterase is an enzyme that will break down the remaining acetylcholine in the cleft. The action potential travels down the motor neuron to communicate the message started in the brain to have the muscle contract. When the electrical impulse reaches the axon end of the neuron, we must convert the electrical signal to a chemical to cross the synapse. Acetylcholine is released from the vesicles into the synapse to bind the motor end plate on the sarcolemma. This changes the membrane so the action potential can spread down the muscle fiber. This occurs by opening the sodium ion channels as the action potential spreads. Opening the sodium ion channels triggers the release of calcium from the sarcoplasmic reticulum. This is important because the calcium is used to remove the troponin and tropomyosin complex from the myosin binding site. Now the myosin binding sites are exposed. With the next two slides, we will show illustrations of the neuromuscular junction. So here we can see we've got this motor neuron here, the sodium ion channels being open, your motor end plate where they bind, acetylcholine esterase coming along afterwards to break down the acetylcholine, your acetylcholine here in the synaptic cleft. And this is another illustration showing the same thing here.